Now that we have established that assembling proteins randomly is not feasible, let's see how living cells actually do it. And this is where DNA enters the picture. All the sequence-specific information required to build proteins can be encoded in DNA. What makes this process particularly fascinating is that DNA itself is not physically or chemically related to amino acids or proteins. Instead, the relationship between DNA and proteins is purely semantic, where DNA is, essentially, a digital data storage medium.
I could fit all movies ever made inside of this tube. So anything, really anything, that can be stored as zeros and ones can be stored in DNA. We just have to convert our data, all of those zeros and ones, to A's, T's, C's, and G's.
It's not just us. The cell also uses its DNA as a versatile data storage medium. The vast majority of DNA doesn't code proteins. Unfortunately, for decades, these non-protein coding parts were viewed as junk by the scientific community. But more in-depth research revealed that this couldn't be further from the truth. These parts play crucial roles in gene regulation, error correction, RNA regulation, epigenetic regulation, and chromosomal stability, just to mention a few. When it comes to encoding amino acid sequences, it's merely an issue of informatics. The task at hand can be phrased like this. How can you represent 20 different kinds of amino acids by using only 4 different characters? Self-evidently, using a single nucleotide would only allow for 4 possible amino acids to be encoded. Using 2 nucleotides would allow for 16 possible amino acids, which is still not enough to represent all 20. Therefore, a minimum of 3 nucleotides is required to represent all 20 amino acids. But 3 nucleotides allow 64 possible 3-letter combinations, so there is wiggle room for using multiple codes for every kind of amino acid. This wiggle room also means that there is an astronomical number of possible arrangements. There is essentially an infinite number of ways to create different codon assignments. From this almost infinite sea of possibilities, the one arrangement chosen for life is very special. This is the universal genetic code of life. This particular arrangement is optimized to the absolute extreme to prevent random mutations having an effect. It is fine-tuned to preserve the initial function of proteins and to prevent change through mutations. It's also optimized around the amino acid needs for protein stability. Considering these aspects, it's estimated that our code is optimized beyond 1 in a billion. We know that this genetic code is the same since Luca, the last common universal ancestor. But to explain its special features, it's theorized that there was a preceding era while it was being heavily optimized. However, this leads to the paradoxical view that this system was optimized by the effects of blind mutations to prevent the effects of blind mutations. Let alone the self-defeating nature of this proposed process, optimizing the genetic code with purely Darwinian mechanisms is technically unworkable. Much like how changing the encoding of a document breaks the text it contains, changing the genetic code would break the genome of the organism. What you see here is an abstraction. This table, in this form, does not physically exist within your cells. Instead, there are molecular agents that are responsible for knowing and applying the rules. Codons are merely symbols. You can place a piece of messenger RNA with a perfect mixture of all 20 types of amino acids into a petri dish and add fully functional ribosomes, but you won't see any proteins forming, no matter how long you wait. This is because symbols alone don't achieve anything. You can give a monkey a map to a lush banana grove. It won't be any help for him. Symbols are essentially meaningless without a system that connects them to their intended meanings. In living cells, there are unique agents known as amino-acid tRNA synthetases that play this role. These molecules serve as the keys for the genetic code, with each type of amino acid having a corresponding tRNA synthetase. However, the task of connecting symbols to their meanings is far from trivial. One end of the molecule must read and recognize codons, while the other end must recognize and attach the corresponding amino acid. What we have here is truly remarkable. We have a molecule that utilizes symbolic representations to reference other molecules, DNA, and an information processing system that comprehends these symbols and links them with their corresponding meanings. The emergence of such symbolic and language-like features in physical matter presents a conundrum that still eludes explanation.
Translation occurs in structures called ribosomes. So the messenger RNA is read off the DNA, and in the ribosome it is pulled through like a tape through a tape recorder, and the sequence of those nucleotides specifies the sequence of amino acids, so this building chain of amino acids then forms the protein.